Thank you, everybody. It's a real thrill to be back. Like I never left. It's great. And I'd forgotten what a lovely auditorium this is, and especially when it's full. So thanks for coming out. This, as always, will be a busy lecture. And I want to talk philosophically. You would have noticed that from my abstract. Paul said he's got this great new series and he wants people to be not just interesting, but provocative. And so I said, well, I'm not used to doing that, but I'll do my best. But I thought I'd just start. I didn't intend to say this at the outset, but I will. You know, I spent 24 very happy years at this university and thrilled with Brandon and Paul and Jenny and everybody else in this audience to build this place into what it is, one of the best institutes in the country. And I went back to the lab to try to prove that people have misunderstood genetics for the last 50 years, and that'll be the main narrative today. And it's that narrative that took me into questioning almost every aspect of the conceptual framework of molecular biology, because every time I looked at something which conflicted with the possibility that RNA might be functional, turned out it was based more on preconception than it actually was on evidence. And this is riddled through, I think, not just molecular biology, but science as a whole. But I'll come back to that. So I went back to the lab as a Federation Fellow, then Australia Fellow. I was very privileged, and we had a beautiful group. Some members, like Mike and Igor, are still here. And I was very happy, but I got tempted to go to Garvan because I thought that it was time to shift gears, and the opportunity presented itself there to move into genomics. So just quickly, and I mean this for the students more than anybody, I'm convinced that, well, there's an old saying, at least my old saying, is that genetics discovers and biochemistry explains. And I think you can look at the whole history of biological research and find that statement to be generally, not always, but generally true. And genomics is a new genetics. So we've just finished sequencing a 5,000th genome. We'll be doing another 20,000 in the next 12 months. And to me, the next period of big discovery in biomedical science is going to be the integration of genomic data with phenotypic data from the medical records, Fitbit devices, etc. And as a taster of this, and you can go online and look this up, Craig Venter's company in California, San Diego, has just announced having sequenced the first 10,000 genomes of their particular cohorts, and matching that to photographs and then using machine learning can actually accurately, and I mean accurately, predict the face of the individual from their genome sequence. That's it. Unbelievable. Now think about that, not only what it means in social terms, but what it means in discovery terms, because if you can do that for three-dimensional facial characteristics, you can do it for a lot more as well. So I think the next generation of medical discovery is going to be centred around individual genetic information, both at a personal level but also at a metadata level, integrated with medical records, and machine learning is going to take over. So that's my philosophical front end. But the narrative I want to talk about today, and I've forgotten it's kind of warm here. Sydney, it's cooling down in May. And forgive me, I've got a cold, which makes it even worse. As I said, this philosophical realisation that most of what we believe and most of what's in the textbooks, conceptually, I don't mean the details of the structure of ribosomes or whatever, but that conceptually is actually both based on very little evidence, it's based on preconception, and it's almost certainly wrong. And I said in my abstract, aided by reductionism. And what I mean by that is that if you are studying a kinase or a transporter on the cell surface or a transcription factor, you're not really you're not really forced to look at the structure of the system. So you can be happily travel along with these background assumptions. Lord Kelvin, in the last part of the last century, about the time that my grandmother was born, announced, pontificated, that there was an end of physics. End of physics. And by the way, I do recommend people study the history of science, and you'll realise what an interesting journey we're on, and we don't know where we are on that journey. But Kelvin said, we've got Boyle's law, we've got Ohm's law, that's it. It's all over. And then along came Rutherford and Einstein and Schrodinger and threw all the cats up in the air. We're still trying to work that one out. The same feeling was abroad at the end of the 20th century with the genomics projects, and it came from the biochemists who really said, gee, that's great, we'll sequence all these genomes, we'll find out all the genes and all the proteins, and we just have to work out what their structure-function relationships are, and then we can put Humpty Dumpty back together again. And I think that was equally, well, it's hubris to begin with, but was equally flawed. And so I want to show you why. And I want to start with one of my favourite, and I have to credit Ryan Taft, and many of you know, for bringing this to my attention. This is Barbara McClintock as a PhD student. Now, she got the Nobel Prize in the early 80s for discovering transposition. I'm going to circle right round to her at the end of this lecture if I talk fast enough. This is her among her Mays, and she got the Nobel Prize. She was the grand dame of Cold Spring Harbor. And in 1950, she wrote these words to her friend: "Are we letting a philosophy of the protein coding gene control our reasoning? What then is the philosophy of the gene? Is it valid?" She goes on to say, when one starts to question their reasoning, she means the logic and evidence behind the origin of the present notion of the gene held by most geneticists. The opportunity for questioning its validity becomes apparent. This was written in 1950, 3 years before the double helix. And because she had intuited, in the best sense of that word, looking down her microscope at chromosomes in maize, that genes just didn't only make enzymes. Now, there's a big war, a cultural war, between the biologists, the Drosophila people, the maize people, and the biochemists who entered the field in the 40s, notably Beadle and Tatum, who coined the phrase one gene, one protein, so one enzyme, which later morphed to one gene, one protein, and had this very mechanical view of the system. And Barbara realised it was wrong, or at least she thought it was wrong. Her views never held sway for various reasons, a biochemist one. I have to say at the outset that I can tell you that almost every conceptual framework in molecular biology is wrong because of that wrong assumption at the beginning, that most genes encode proteins. That's true in bacteria, but it is not true in humans. So the assumption, based on limited studies like Lacopera and E. coli, and of course bacterial genomes are wall-to-wall protein coding sequences, has been, and everybody in this audience would accept this, it's imbued in every bone of the textbooks, that genes make proteins. Of course, there's a few microRNAs here and a few transfer RNAs there, but basically the system is a mechanical system run on proteins. And that most genetic information, including regulatory information, this is a bold of that for a reason you'll see, is transacted by proteins. Now, I've been around long enough to understand, been around 2 or 3 blocks in science and molecular biology, and when you look back, you realise that the foundational assumptions of molecular biology, which are set down in the 50s and 60s, within a decade of the double helix, 53 to 61 was the Lacopera, really reflects the deeply mechanical and reductionist zeitgeist of the time. Now, zeitgeist is one of my favourite words, like Schadenfreude. These Germans have wonderful words. Zeitgeist means the prevailing cultural paradigm. The prevailing cultural paradigm at the time was that it was a deus ex machina, that genes made proteins and the system was mechanically made of proteins. Now, of course, we know proteins are important, so don't get me wrong, but this protein-centric view has led to several subsidiary assumptions, despite a number of subsequent surprises that should have given pause for thought. Somebody once said that the best science is done at the point of greatest surprise. And if that's true, and I think it's a good saying, then molecular biology has failed that challenge 3 times. It's almost like the 3 bells, you know? And the first, and that's why we got interested in this, is that the genes in humans and other complex organisms are mosaics of protein coding and non-coding information. Now, the interpretation, the immediate, and this was in the late 70s, the universal immediate interpretation all the way from Crick was that, well, everyone knows genes make proteins, right? So this stuff which is transcribed but not translated must be junk. And it was rationalised as a hangover of early evolution, fattened out by transposition. That ended up in all the textbooks without a shred of evidence. That was just one of 2 possible explanations. The other possible explanation, which occurred to me in a pub in the Texas Medical Center late on a Friday night, was that maybe the RNA which is being transcribed but not translated is itself transmitting some information to the system. Now, that alternative hypothesis, based on a very limited set of facts, is not only equally valid at that point in time, but has the great advantage, at least I thought so, of being interesting. Interesting. So, and I stuck with it. Now, the second surprise, about the same time, and I hope at the end of this, I'm not sure if I'll convince everybody or it'll even upset you, but that you'll see how primitive the current conception of the genome is. So the second surprise, about the same time, late 70s, was to discover that our genomes are full of retrotransposon, transposon-derived sequences. And on the same dodgy logic, these things were condemned as mainly non-functional, selfish DNA. as I'll show you later, they're not only agents of change in evolutionary time, but they're agents of change in real time, particularly in the brain, but also to some extent in early placental development. And the third surprise more recently with the genome projects is that the numbers of genes doesn't stay with developmental complexity. And I remember the time, it was Bob Tijan, I think, wrote an article in Nature or Science where he mooted on this surprising observation and basically said, well, you know, given the combinatorial control of gene expression, we get explosive possibilities. And that was it, one sentence, and then went on to go talk about just so stories in gene regulation. Let me say disrespectfully, in my entire life in molecular biology, I've met many people who work on gene regulation, but I've yet to meet anybody who actually knows anything about regulation, who's crossed the road intellectually into decision theory or control systems theory to actually understand how systems might be regulated, as opposed to this protein binding to DNA at this consensus sequence. I'll get more provocative as we go on, but let me pick this up. What Bob Tijan, a very fine biochemist, by the way, and I think Rick Sturm, who used to work here, did his postdoc with him at Cold Spring Harbor, was hinting at, but didn't formalise mathematically, was that the tacit assumption that if you have combinatorial control of gene expression, then you have a factorial expansion of possibilities. In other words, if you've got n regulatory factors, you have n factorial or n prime possibilities. So if there are 3,000 regulatory proteins in the C. elegans or human genome, and there's a minimum of that many, then 3,000 factorial, I can assure you, is a number that's almost infinitely, at least figuratively, infinitely bigger than numbers of molecules in the invisible universe. More than enough regulatory power there, boyo, to program C. elegans of humans, so we can happily go back and study our favourite transcription factor, safe in the knowledge that combinatorial control will explain the difference between these two organisms. Now, the worm on the right, C. elegans, you're very familiar with it, 1,000 cells, few muscle cells, few nerve cells in the gut. One on the left, between 10 and 100 trillion cells, myriad of muscles and bones and organs and a brain of 100 billion neurons, each with an average of 10,000 connections that can walk and talk and remember. Bloody hell. Same number of protein coding genes. Just under 20,000 is the latest assessment. What's worse is that most of them are orthologous. So the Lego part set of human development or animal development hasn't changed much in 700 million years. Half of all the proteins in your brain are actually have orthologs in sponges. Right, okay. So, but, you know, it doesn't matter because we know combinatorial control will explain. But the question is, and I don't think developmental biology has ever asked this question in a structured way, is how much information is required to program human development and cognitive capacity, how does it scale, and what form does it take? Now, it's a cartoon I like to show. I did in Japan recently, so I changed it to Toyota. It makes the point, I think, explicitly that if you're going to program the ontogeny of a complex object like a motor car, you can specify the component set, think of this broadly as the proteins, and look at their structure-function relationships. And I'm not meaning to demean this because it's really important that we understand what each component does and how it does it in four-dimensional space. But you also need another set of instructions to put this together. And anybody who's taken home a bookshelf from IKEA can know how challenging that is. Imagine taking this home. You know, you have books carting. So the question we asked some time ago with Larry Croft and Michael Gagan here at UQ and Martin Lurcher in Bath was how do regulatory genes scale with complexity? Some of you have seen this graph before, but I think it's a really important one. So this was done some years ago. We repeated it more recently with thousands of genomes. It doesn't change. The graph on the lower right is the primary graph. The main graph is the log-log transformation. And those of you who are mathematically literate would know that when you do a log-log transformation of the data, you get the slope as the exponent. On the x-axis is the number of genes. This is in bacteria where you can actually fairly accurately decide what's regulatory and what's not by, you know, say, computational analysis and PFAM domains like helix domains, etc. Regulatory proteins on the y-axis, total on the x. And it scales it almost precisely too. It's a quadratic relationship. Don't know if it's n log n, and one of my unfulfilled challenges is to try and reconcile this with decision theory, so you've actually got a mathematical basis for this empirical relationship. This is exactly the opposite of what's predicted by combinatorial interactions. I don't have time to go into that, but it's exactly the opposite. But basically what it says is as the system gets larger and more complicated, more and more of the information in the system is devoted to its organisation and ontogeny. And the top end of this graph, because you're probably aware that bacterial genomes top out about 10 megabases, over 20% of all the genes are regulatory, and it's going up quadratically. So if you extrapolate that straight line, and the R squared value is 0.9, it's very good, then you get to a point somewhere up there where there's more regulatory genes than genes, which is clearly impossible. So this is a very plausible reason why bacterial genomes have had a limit imposed on them, because they can't grow anymore because the cost-benefit analysis is not worth it, and which means that the eukaryotes must have solved this problem in some other way. That's another lecture entirely because I have to move on. What you can show empirically, and there's facts and interpretations always, is that the proportion of non-coding DNA, which was the regulatory sequences are located, broadly increases with developmental complexity. And this is an old graph that I did with Ryan Taff some years ago, and we updated more recently. It hasn't changed. Basically, bacteria have some low amount of these sequences, 5', 3' regulatory sequences controlling transcription and translation. The eukaryotes get well up above 25%, even Saccharomyces cerevisiae and Plasmodium, then the fungi and the plants, and up to humans at 98.8%. Moving quickly, it's now evident, and I'll show you a bit of evidence, but take it as read, that irrespective of the extent of these non-protein coding intronic and intergenic sequences in the genomes of eukaryotes, they're all transcribed. They're all differentially transcribed. So it's not just introns being transcribed in RNA, the whole system is transcribed differentially. So you force the conclusion either there's an enormous amount of useless transcription, riborecycling, not just in introns, but antisense RNAs, intergenic RNAs, etc., or there's another layer of genetic information transacted by RNA that expands in complex organisms. Now, when I first entertained this idea, and one of the reasons I got sort of narky about this is because the establishment said, oh, it's up to you, boyo, to prove it. And I thought, well, okay. And then I realised that the existing things in the textbook had no evidence. And then you realise that in science, and this is something you want to keep in mind, I'm talking mainly to students now, because I can tell you with some conviction that what we did in the 20th century was just the foothills. This is the coming century, and you need to have a free head. In science, if you make an observation, it's natural, and we all do it properly, to try and generalise that observation. But if you can't test the truth of that generalisation, it sits there as the accepted explanation, then becomes the orthodoxy, then becomes an article of faith, with no more evidence than it had for original speculation. You come along with a different set of interpretations, or facts that might conflict, or interpretation might be different. People say, oh, it's up to you to prove it. So I accept that criticism for a while, but I don't anymore. I think it's up to everybody to reassess. And I can tell you from my experience in science, I hope there's not too many people like this in the audience, that when you tell them something is unusual, many people in science will fold their arms. Prove it. Well, they're not going to discover anything. The good ones will lean forward and say, tell me more. Tell me more. Doesn't mean they believe you, but they're interested in the fact that you've got a different take or a different set of observations that might cause them to reassess the way they think about the system. And going back to Lord Kelvin, the reason I've introduced him is that the idea that our conceptual framework at the end of the 20th century is anywhere near complete or even accurate is just as stupid as Lord Kelvin at the beginning of the 19th century. Yet we all happily travel along and don't actually challenge the conceptual framework in which we work. We say we're interested in human biology. We say we're interested in the function of the brain. We say we're interested in how this biology transacts in disease. But we're not actually interested in how the system works. Or at least don't go to try and study it. Moving along quickly. The data for this has come out over many years through David Hume, who many of you know well. We were involved in the Phantom Consortium at Riken in Yokohama. This was high-throughput RNA sequencing a decade ago, motivated by trying to define the proteome and the spliceome. But the unexpected and ultimately headline result was that tens of thousands of RNAs being produced that had little or no protein coding potential. Not just intronic RNAs, but lots of other ones. Same time we showed there was 70% of all mouse genes had overlapping antisense transcripts. Presented the first evidence that this might have some regulatory function. Tom Gingeris showed the same thing about a few months earlier. And I just mentioned that, for what it's worth, that almost half of all human transcripts are not produced by POL2, and they're not polyadenylated, and they come under the radar. This discovery is being forgotten for the second time. And more recently, this paper in Nature a year or two ago showing that only 5% using conventional signatures of promoters, this is chromatin modification, methylation, modification signatures, etc., only 5% of transcription start sites in the human genome actually produce RNAs that produce proteins. So about 20-fold ratio, and that's our estimate too. So if there's 20,000 protein coding genes, there's something of the order of 500,000 non-coding RNA genes being produced. The whole thing's alive. And it's amazingly complex. I think in the interest of time, I'll skip past this. Igor and I wrote this 2006. And this cartoon we drew back in that review, even then was an underestimate, oversimplification of the actual landscape. So I want to show you some recent works that just brings home how complex the transcriptome is. I sometimes title lectures, The Human Genome is a Zip File Extraordinaire, because the transcriptome is at least 2 orders of magnitude more complex than the genome. And it gets unzipped in different patterns in different cells at different stages of development. So through Tim Mercer in conjunction with John Rinn at Harvard, we developed, and Tim used to be here as well, we developed this sort of molecular microscope for transcriptomics, where you just capture the transcripts from the region of interest on oligonucleotide arrays. We use beta arrays now, and then sequence. And I'll just show you what the first data, this was published a couple of years ago now, but in this graph, this Circos plot, are regions of the human genome in fibroblasts that had little or no transcription evident in conventional deep sequencing. So just look at this lower left one here. There's nothing here. That's what you see with conventional deep sequencing. On exactly the same prep, we captured transcripts that might have come from that region and then resequenced. And this is what you see. They're alive with transcripts, and each one of these arches is a splice junction. So not only are they transcribed, but they're also alternatively spliced. So things that look to be transcriptionally silent gene deserts are actually alive with transcripts, which means there's a subpopulation of cells in this system, this fibroblast in culture, which are in different transcriptional state. And this is protein, and we did the same thing with P53 and Hoxlow, so I'll show you those in a moment. On the outside, what we got with conventional sequencing, and the inside here in blue, all the additional complexity, both coding and non-coding, splicing patterns, etc., that you see with a molecular microscope. So here's one of the lessons of this lecture. Averages lie. And I'll show you this in finer detail shortly. But basically, much of our studies on differential gene expression is predicated on the assumption that when you take a sample of kidney or whatever, that what you see there is representative of the population rather than a smudge mixture of subpopulations. What's also true for this is that all of the statistics around microRNA analysis are predicated on having basically a one-dimensional Gaussian distribution of gene expression patterns. But in reality, it's an n-dimensional bimodal distribution. And you don't know when you see something go up, or two things go up, whether they're going up in the same cells or not. And David Hume first pointed this out some years ago when he showed that, if you induce beta-galactosidase in E. coli or HeLa cells, the cells go from white to medium blue, dark blue, and you think it's like a rheostat. We always think it's like a rheostat. But it's not. There's only two sorts of cells under the microscope. They're either white or dark blue. And so as you induce these gene expression, you have a larger proportion of cells that switch into a different state. This comes back in ways that I don't think I can explain to decision theory, but you can make a first approximation statement, although in time it will get fuzzed, that everything in biology is a binary system. So let me just, in this one experiment, just to keep moving, we discovered 4 new isoforms of P53, which is probably the most intensively studied gene in human biology. A bunch of new Hox transcripts, both coding and non-coding. And this non-coding locus here, which only showed 3 exons and 2 splice junctions in the original deep sequencing data, when you drilled into the locus, you saw this extraordinarily complex pattern of alternative splicing. I've taken it out. We've just done a high-resolution analysis of chromosome 21. What we find are 2 stunning findings. One is that the level of alternative splicing is about twice as high in human genes as is in mouse genes, and this is a high-resolution analysis. And also that in long non-coding RNAs, which I'm mainly talking about, are like this, that they're almost universally alternatively spliced at much higher level than protein coding transcripts, which implies that the structure of these RNAs is highly modular. But I took it out in the interest of time. You'll be grateful as I keep going through this. We've also done with the genome-wide association study. Some of this has been done with Grant Montgomery. Grant's up there somewhere. You know about these things, so I won't explain it. But just say, basically, from a genetic point of view, and this has misled people for a long time too, if you make a mutation in a protein coding sequence, you have what I call catastrophic component damage. You don't have the chloride ion transporter, you've got cystic fibrosis, you don't have a hemoglobin, you've got thalassemia. You're in big trouble if one of your core components is not working. So the phenotypic spectrum for protein coding mutations is very strong. And you can say the first approximation, we're finding this with our whole genome sequencing in humans of kids with disabilities, that about 95% ballpark of mutations that give you severe genetic problems are protein coding. But you see the opposite ratio with variations that underpin complex traits, but nearly all intronic or intergenic, which means, and nobody's surprised at this, that these are more regulatory and they're affecting the architecture of the system rather than messing up the components. So we set up, this was under, with Marcel and Dinger, who you know as well, with Nina Bartonesek, a great postdoc at Garvan, and we captured a whole bunch of disorders, covering 2% of the genomes. I'm moving quickly through this, but an extraordinary landscape, and I'll just show my favourite from this is a haplotype block. Now, you know with GFS studies look at haplotype blocks association with particular conditions. So here's the haplotype block in red. You can see down here there's virtually nothing in the database. There is a protein coding gene next door. This is the sentinel SNP that's used in the analyses, and this is here is a non-coding RNA gene that's alternatively spliced that sits right in the middle of this haplotype block. And it's got to be a very good candidate for the molecular basis of this particular phenotype of cognitive performance. This is another locus. We found 3 non-coding RNAs in this haplotype block. And just to summarise, we've got at least 1,500 new genes that are sitting right in the middle of so-called gene deserts, RNA genes, that are associated with complex diseases. These non-coding RNAs are extraordinarily show the same range, I should say, of stabilities as protein coding sequences. It's a log scale in this graph. There's incredible variation, both in proteins and non-coding RNAs, as to whether they're stable or not. So these things look pretty like regular RNAs and genes. The genes encoding these things, and I'm smudging a bit just in the interest of time, but they show all of the features of being real genes. And I won't go through this list, and I published it some time ago now, but for example, they show exactly the same features of chromatin signatures, epigenetic signatures that messenger RNA genes do. They're polyadenylated, many of them, though many are not. They are alternatively spliced. The expression is regulated by conventional morphogens and transcription factors, and they show very, very precise expression patterns. This is probably the most important slide, and it's my favourite. Funnily enough, when we first published this, we had a lot of trouble getting it published, and this is work that Tim and Marcel did in my lab back here in IMB, published in PNAS 8 years ago now. Its citation rate just keeps going up and up and up and up, because people are finally realising what it actually says. So this is in situ hybridisation analyses of about 1,000, or over 1,000, of the recon RNAs that we identified, done in collaboration with the Allen Brain Institute in Seattle, because they had the muscle to do it. And the controls on the left, this is the primary data, this is a computer transformation, which gives you better contrast. I hope you can see this clearly. But if you look at panel B here, this RNA, as far as we know, is only expressed in the dentate gyrus of the hippocampus. In panel C, the RNA there is only expressed in the CA1 subfield of the hippocampus. Now, if you mashed up these brains and did deep sequencing, you'd be lucky to see these things, only expressed in a limited number of cells. Right? Averages lie. These things, and one of the criticisms of this field was, and Mike, I had to take it out in the interest of time, your graph about the conservation of circular logic. One of the furphies in this field is that most of the genome is not conserved. That's an incorrect statement. By the way, I'll just say what you and I worked out years ago, is that the accurate statement about conservation of the human genome in respect to other mammalian genomes is that most of the human genome is evolving at the same rate as ancient retrotransposons, which have been commonly in the mammalian lineage for the last 300 million years. So on the dodgy circular logic that the ancient retrotransposons have been in all our lineages for 300 million years and non-functional, big assumption, then you can say that because the rest of the genomes are evolving at a similar rate, that must be also non-functional. If the assumption's right, so is the conclusion, but vice versa. The other criticism has been that these long non-coding RNAs, which were totally unexpected in these data sets, were expressed at much lower levels than protein coding genes. That is incorrect. They're actually expressed at very high levels. but only in very particular cells. And in fact, I couldn't pull it out, but when you plot the numbers of cell types that different protein coding genes are expressed in, you see most are expressed in many cell types. You see exactly the opposite graph for long non-coding RNAs, that they're only expressed in very limited number of cell types. And the first superficial interpretation is that these things are defining the cell state in far more precisely than protein coding sequences are, although there are exceptions in both directions. You see the same thing in the cortex and the cerebellum. I wish I could show you this at high resolution. Yeah, if I could show you the cerebellum, I could show you that a messenger RNA gene is specifically expressed in the Purkinje cells, and then in a single line of cells underneath it, the overlapping antisense RNA is expressed just in the single line of cells underneath. Oh, it's beautiful. Look like noise to you? This is very precise. And that's the integrated data. I'll keep moving. Most of them actually show, this is Purkinje cells again, a surprising number, 30 or 40% go to the cytoplasm. There's a whole new field of cell biology here which is just starting to hit the journals. People are starting to chase these things and finding subcellular domains that are being organised around RNAs in the cytoplasm. Most of them are nuclear and chromatin associated. We'll get back to that. Others are decorating weird subnuclear organelles. This is in Purkinje cells. We thought these were centrosomes, and I work with Bodo Lang in Berlin, who writes the reviews and so on. They don't co-localize with centrosomal markers. So there is a as yet unknown subcellular structure that, as far as we know, only exists in Purkinje cells. We have no idea what it does. These are paraspeckles, and they're in neurites. So with paraspeckles, with David Spector, we showed these things, long non-coding RNAs, essential structural and functional components of these things that are very enigmatic. And in fact, if you knock them down, you get very little phenotype, although they're very widely expressed in differentiated cells, but you get placental phenotypes. And one of the big mysteries, and there's mysteries everywhere here, is that, by the way, and the biggest one is MALAT1, a non-coding RNA, which is the most highly expressed gene in the human genome, leaving aside ribosomal RNAs. It's more highly expressed than actin. Knock it out, no phenotype. It's vertebrate-specific RNA. Coats the chromatin. You do get increased susceptibility to cancer and some weird other phenotypes, but there's something going on here below the hood that means that some of the most highly expressed genes in the human genome, or the mammalian vertebrate genome, do not appear to have an overt developmental phenotype. And my suspicion is it's got more to do with this than it does with this, but we'll see. Another one's Gamafu. Guy Barry's in the audience, worked on this with me and others, and with Ernst as well. Was first identified by Shinichi Nakagawa as being decorated novel nuclear domain subset of neurons, and together with Ernst and Seth Blackshaw at Hopkins and Shinichi in Waco in Japan, we showed that this is acutely regulated response to neuronal activation and shows the same alternative splicing effects that you see as a typical phenotype in schizophrenia, and this is physically associated with proteins like quaking-1, which are also implicated in schizophrenia. Over the last decade or so, mainly here, we've looked at the differential expression of these things not just statically in the brain, but in different differential systems, cell differentiation, neuronal cells, T cells, etc. I'm going to show you two vignettes from memory development and melanoma. And we did this by differential display. I'm just going to move through this quickly. This standard sort of stuff. And I'm going to show you the second one, which is an antisense RNA, which is nearby but not overlapping a protein coding zinc finger transcription factor on the other strand. And this thing shows high expression in pregnant mammary gland, but it's turned off in lactation, then turned on again at weaning. Keep that in mind. It's off in lactation. So it's very stable, this RNA, which is unlike the protein coding transcript on our strand. But if you knock it down, and it's very easy to knock these things down with siRNA strategies, you see that compared to a scrambled control, when you knock down this non-coding RNA, you increase the proliferation rate of mammary epithelial cells in culture. Remember, it's down in lactation. Knock it down in culture, the cells grow. They also produce large numbers of domes because these mammary epithelial cells in culture, when they get to confluence, produce three-dimensional domes. So when you knock this thing down, remember it's down in lactation, you get more domes and they're bigger. But the kicker is that they start producing milk, and this is casein. So this is the mammary epithelial cells in a control. You knock this thing down, it's turned down in lactation, these start pouring out casein. So we can turn mammary epithelial cells into milk-producing cells in culture simply by knocking down this RNA. Melanoma, same thing. We look for things that are differentially expressed. I'm going to focus, I'm going to move quickly through this one, which is a smaller RNA, which is produced intronically in this protein-coding gene in the other direction. A couple of quick points. One is it's not conserved. Look at this conservation peak. This is all mammals. Exons, of course, are conserved because they're primary and very stable, Lego part components. But this thing is obviously an important feature of this intron, which has got pan-mammalian conservation. This one's only conserved in primates. Quick point. Conservation is a crock. If you have conservation, it imputes function. Lack of conservation imputes nothing. And regulatory sequences evolve very quickly because they have different structure and function constraints than hemoglobin or a wheel, analog devices. Let me put this to you. You see very low conservation of promoters in front of genes. Very low at the sequence level. But nobody says they're not functional. It's just that their structure function constraints are quite different. And they're also subject to adaptive radiation positive selection for phenotypic variation. I'll bet many of you, if not most of you, don't have the same phone number or credit card number that you had a decade ago. Doesn't mean that that piece of information as a digital string isn't important. So you have to think very carefully about this. This one's not conserved, and it also folds very high-level prediction into this three-dimensional structure, which reminds me to tell you that RNA is not a wiggly line with A's on the end of it. It's a very sophisticated three-dimensional molecule with hydrogen bonds on all three faces, Watson-Crick face, the Hookestein face, and the ribose face through the 2' hydroxyl. It forms these structures, and this is why this is RNA's sweet spot and why this is central to all of biology, because it not only has these folded domains in 3 dimensions, not just 2, that can analog structures that can bind proteins, etc., but also these open faces that can interact with other RNAs and DNA. So the big strength of RNA is it connects the analog and digital worlds, and this is what it's doing in biology. So if you knock this thing down, this is remember it's up in melanoma cells. With different levels of siRNA, you can reduce the invasiveness of these melanoma cells. Here's the quantification. This is small enough to actually knock it up, and this is the gold standard experiment in long non-coding RNA biology, not just to knock it down, but so when you knock it down, it's up in melanoma cells. Knock it down, it reduces their invasiveness. If you increase its expression on the control on the left and the overexpression on the right, and you can see if I run that movie again, that the ones on the left are much more aggressively motile and invasive than the ones on the right. And this, along with another one that John Rinn discovered, is probably the most powerful gene yet associated with invasive metastasis yet discovered well, that may be a too bold a claim at this point, but was certainly one of the first genes to be shown to be involved in controlling cell motility and invasiveness, and there's no way in the world this is encoding a protein. A couple of things I spoke in Cleveland recently. I pulled them out just to nod to my host, but there's now been several hundred papers showing that if you knock down or muck around with these RNAs, you see biological effects. And here's just a couple of cases of reprogramming mesodermal cells into neurons and regulating gene expression and methylation patterns in colon cancer. Now, the clue to what they do, they're doing many things because the ones in cytoplasm are clearly doing something different from those in the nucleus, but it was from this experiment we did with Marcel and Paolo Amaral here some years ago. We looked at differential expression of this during ES cell differentiation in culture. There's only 2 protein coding genes here. This one, this transcription factor, this is primitive streak formation. There's hundreds of non-coding RNAs going up and down by orders of magnitude during these transitions. It's a messy transition, but nevertheless, I'm going to concentrate on this one, which finally got to publication recently with Perco in the audience. And it was Paolo first identified this thing as it's a non-coding RNA that lies antisense to this homeotic gene EVX1. And we also looked at this antisense RNA which lies right across on the other strand the HOXB5 and B6 genes. Now forget all this detail. What Paolo showed, and it was an inspired experiment on his part, was that these antisense RNAs opposite these homeotic genes are physically bound to trithorax, that's mixed lineage leukemia 1, and modified histones which are indicative of active chromatin. The messenger RNA is not bound to these things. This one is, but this one is not, and is probably bound to polycomb, another epigenetic regulator. So this is one of the first evidence that showed that these RNAs were associated with chromatin-modifying complexes. Now, I probably don't need to tell this audience this, but well, part of it I do. Epigenetic processes are often emphasised because of this middle thing here, the long-term responses to environmental variables, key factor in the etiology of cancer, type 2 diabetes, etc. But they're essential for differentiation and development. They first discovered in Drosophila, knock out polycomb or trithorax, you screw up development. And they're essential to brain function. If you don't have epigenetic processes, you can't learn. Now, as you well know, this epigenetic information is embedded in the modifications of nucleotides in DNA, and I'll come to very shortly in RNA as well, and a very wide range of modifications that modify the histone tails in nucleosomes. Now, these modifications are catalysed by a suite of about 100 enzymes that literally impose these chemical marks on the histones or the DNA at literally millions, not just hundreds of thousands, millions of different locations in different cells at different stages of differentiation. If you look at the ENCODE data, in every cell line, the patterns are different for this histone modification or that one. But these are 100 enzymes. Back to Barbara McClintock. These are just enzymes. So clearly there's another level of information required to determine where they compose these marks in the genome at which cells, at which stage of development. Now, the background assumption has been, it's transcription factors. Right, it's transcription, we can forget about that. But one of the funny things, and Garvan's pretty good in this area, about epigenetic mapping of cytosine methylation, for example, is that hardly anybody in that field actually thinks to ask why it changes in cancer. What are the pathways to make it change in cancer? They look at the effects of it, silencing loci as surrogates for knocking out tumour suppressor genes or whatever. So the question is, what determines the site selectivity of these enzymes? What determines the positioning of nucleosomes is important. It is. And what's the molecular basis of environmental epigenome interactions? And the answer to all three of these things I would submit to you is RNA. And this is a general explanation for this massive hidden layer of information that's pouring out of the genome, previously considered junk, but actually differentially expressed. And I've shown you that in the brain in spades, but it occurs everywhere. And we weren't the only ones to show this. About the same time, people working in printed RNAs, printed loci, this one's very famous. This one's 105 kilobases in length, the encoding RNA. Epigenetically silences transcription by targeting polycombs, G9A, same as this group from Kanduri in Uppsala, Peter Fraser's in Cambridge. And then a year or so later, our friend John Rinn writ this large by looking at 3,300 of these things, as only the Broad Institute can do, and showed that 20% of these RNAs are bound by polycomb. Additional ones are bound by other chromatin-modifying complexes. You knock them down, you see the expected relief of repression you would with polycomb relief. So this writes large what we'd shown in isolated settings. There is no question, and my guess is 40 to 50% of these RNAs are actually physically involved in guiding epigenetic complexes to supervise the four-dimensional ontogeny of humans. It's not just transcription factors. In fact, while you can trump these programs by ectopically producing expressing a transcription factor like MyID, that's not the way it occurs in vivo. There's a four-dimensional program, starts off with a single cell and produces this, and does it very accurately, because if I have an identical twin, then I have phenocopy. Every muscle's got MyID on it, and you put a MyID into a hepatocyte or a fibroblast and say, yes, sir, I must be a muscle cell. But every muscle in your face and your fingers and your hands is different architecture. It's not just cell type differentiation. That's relatively easy to program. It's the architecture that's hard. And you have to do this in four dimensions, so you put fingers and toes in the right places. So, moving along, this was written some years ago, but nothing's changed except more data since then. But it's obvious that the whole of the chromatin architecture that epigenetically supervises our expression is being controlled by RNAs that take chromatin repressive complexes, chromatin activating complexes, to organise the chromatin. And I'm going to show you in a minute, this is... No, I think I took it out. But it happens in three dimensions as well. So, and it also involves small RNA pathways, and I don't have the time to go into it, but there are argonauts in the nucleus. There's a whole network of transactions between small RNAs, piRNAs, microRNAs, etc., organising this. And then more recently, Tim and I wrote this review, just elaborating the structural, because there's more evidence now. And so RNA can bind to other RNAs, it can bind proteins, it can bind DNA. I'm going to show you a spectacular picture in a moment. It can act as conformational switches. It can then, in biological terms, be a sort of bring complexes as scaffolds into big transcription and splicing complexes, or guides to guide epigenetic modification. I took it out in the interest of time, but Tim and I, with John Mattick in Seattle, showed in Nature Genetics last year that even alternative spliced exons are pre-organised with the promoter in exon-specific and cell type-specific ways. So even alternative splicing is an epigenetically controlled phenomenon. The cells know which version of the protein they want to produce in the circumstances. It's not controlled by splicing factors. You knock them out, you get bad splicing. It's actually ultimately controlled epigenetically. So this is a picture, one of my favourite pictures, taken by a Japanese microscopists a few years ago. Hope you can see it. I should turn the lights off. This is antibodies, monoclonal antibody against triplexes in interphase nuclei of human fibroblasts. It looks like the Milky Way. There are hundreds, if not thousands, of locations across the genome where RNA is complex with DNA and triplex. We've just finished ChIP-seq analyses using RNA-DNA hybrid antibodies, and somebody else just published as well. There are thousands of places around the genome where you find RNA-DNA hybrids, and these are not the sites of transcription. It's a very interesting pattern. So we're starting to get the first real evidence that RNA is actually physically interacting with the chromatin. And then you've got all this evidence coming out about enhancers producing non-coding RNAs, and the first incipient evidence that the chromatin looping was required for enhancer action, which is central to development, is actually guided by the RNAs, because if you knock down the RNAs, you don't get the enhancer. And the final experiment hasn't been done yet, but it's to show by chromatin conformation capture that you don't get the looping as well. But it's very plausible, and I think the evidence is now pretty good, that all of the chromatin architecture organisation, not just chromatin modification, but the three-dimensional architecture, is all being built on RNA scaffolds. So one of the big questions, I think, for those of us interested in both gene regulation and human ontogeny is what's the structure or the four-dimensional decisional hierarchy controlling human development? And this is more a hope than an expectation, but do different types of regulation occupy different levels in this hierarchy? For example, is cytosine methylation superior to, in cytosine, to histone modification, X, superior to transcription factor Y, zinc finger, versus KH domain practice, versus a splicing factor or microRNA? Because if it turned out that each of these different chemical modifications occupied a different layer in the decisional hierarchy, then we could make much more sense of what we're seeing at the detailed level about microRNA control or transcription factor controller, because we'd have a framework to put all this massive phenomenological data into some sort of structure and order and start to actually understand how the system's put together. Now, very quickly, a couple of big mysteries, and I'm going to tell you at the outset that neither of the things I'm going to show you now can be explained by any current conception of molecular biology. Any. First is, and Tim did this with Dagmar Wilhelm, and many of you remember, she's now at Monash. I think she went to Melbourne, didn't she? Yeah. Terrific postdoc here with Coops, who came to see us one day and showed us this picture of in situ hybridization of these probes against this gene by Adam in testis. And basically, to summarise very quickly, the coding sequence probe showed, and it's not obvious from this picture, but hybridization in the cytoplasm of the interstitial cells in the testis. But the 3' UTR probe showed a totally different pattern, and here's a blow-up. It was very intensely staining in the nuclei of German Sertoli cells. What the? Now, the guys in the lab, I think, including Mike, and certainly Evgeny and others, have been coming and saying to me for years, there's something funny going on with UTRs, because we look in the browser, you see all of these sequences sitting over the UTRs, but not so much over the message. Now, we know the UTRs connected to the message because they were originally cloned that way. And I thought it was just reverse transcriptase having some trouble getting into the coding sequence, whatever. But this is strange. And it's not just here. We had lots of other examples. Here's another quick one. This gene, which I can never pronounce, here's the coding sequence probe. This is the hippocampus. I can tell you it's invisible. There's nothing there. But it's blazing in the CA1 subfield in the dentate gyrus, the UTR. Blazing. And we had first or second level evidence that at least half of all genes, protein coding genes in the mammalian genome showed this phenomenon. Now, it's not separate transcription, because if you look at the indices of transcription start sites like POL2 occupancy and these classic bimodal patterns of histone methylation, conventional promoters in blue show these patterns, but these, the 5' ends of these what we call UTR-associated RNAs, which are capped, and there is a cytoplasmic capping enzyme. They flat line for all of these indices. Whoops, sorry. They flat line for all these indices. So these are not separate promoters. But they are differentially cleaved from some precursor, because here in the brain, this is, what's this one? This is CAMK2A. In the cortex, you've got these two positions where the 5' end of these RNAs is, and this one is in the hippocampus. This is in Drosophila, and it's the same cleavage site in both old and young females, but I want to show you the genetic evidence now, because this is just molecular. It's like, wow, well, what's going on here? They're differentially expressed, differentially cleaved in different tissues. Now, here's the paper that really, when we started to get back, that blew our socks off. And this is Anna Frusi. Some of you might know her as Embl. Paper, what's about a decade old now. Again, many of the most interesting papers just get ignored. This one's stunner. She shows, or her group shows, we show that expression of the Oscar 3' UTR is sufficient to rescue the oogenesis defect of the null mutant. So when you knock out Oscar, very famous gene in Drosophila biology, you lose oogenesis, among other things. Put the UTR back in on its own in a null mutant that can't make the protein, you recover oogenesis. And then we dug back further, and we found, this is Helen Blau's lab at Stanford. This is alpha cardiac actin, tropomyosin, troponin, and showed that this was a transacting tumour suppressor function. This is in, of all things, ribonucleotide reductase, and in prohibited in breast cancer. And basically, in every case, and there's limited numbers of genetic evidence because people don't do experiments like this except accidentally, you can show that in every case documented that expression of these 3' UTRs induces differentiation and reverses a tumorigenic phenotype. And then this paper came out just recently, a few months ago, in Neuron, showing, and we'd predicted this in the brain because we had lots of pictures of the brain, but confirming what we'd found, that you see this massive differential expression of UTR sequences in neurons and other tissues. So, you want to explain that? Not just the fact of it, but the genetics of it. It changes the behaviour of the cells. This is completely dislocated from the protein coding. So it's not a platform for regulatory protein binding or microRNA binding, it's something else. Now, you can speculate it's a sponge. I don't think it is. I think that's too easy an explanation. But there's something going on. And in vertebrate evolution, you see the 3' UTR get bigger and bigger and bigger and bigger till in mammals, it's as big or bigger than the protein coding sequence, and often more highly conserved. Kilobases. What, landing pad for hundreds of microRNAs? What? So something's going on here which does not fit with our conventional understanding. Here's the other one, and Mike did this. Glad you're here, Mike. It's great because I've brought this to the fore more and more. Eric Westhoff and I decided this was the biggest mystery in molecular biology. Eric being one of the great RNA biologists of the world in Strasbourg. So Mike and we did this at the same time as David Hausler, and I was talking in California, and David came up to me afterwards and said, "Oh, we got the same data." I said, "David, let's not kill each other. Let's just publish it together." So basically what Mike found, by simple expedient of looking for sequences 200 base pairs or more, they're absolutely conserved between 3 mammalian genomes, which only 3 had at the time. And this is, I remember when we wrote the letter to Science, the covering letter, we said, this is a bioinformatic paper. Like, don't ask us to do an experiment. I mean, this is just, nobody expected this. There are hundreds, I'll show you in a moment, thousands of these sequences which are essentially frozen in the mammalian lineage. Thousands of them. And they're all non-coding. And I said to Eric, well, I think, we were discussing this in Strasbourg last year, I said, well, I think 2 of them are in the ribosomal locus. Remember, Mike? And I said, well, you know, ribosomal RNAs are very heavily constrained by multilateral protein RNA interactions, so I assume the rest of these are similar sorts of things. He said, you're wrong. I said, what do you mean? He said, they're not in the ribosomal RNAs. They're in the intergenic sequences between the ribosomal RNAs. And he said this, considered, he's an RNA biology expert. In RNAs, you get covariation in the stems and so forth. He said, I cannot think of any explanation, either in evolutionary space or molecular space, that would explain these things. And with Mike and Stuart Stephen, we went on to show that these things, there were thousands of them. Just quickly, this is coding sequence evolution through the vertebrate lineage from Fugu through the humans. And this is the ultra-conserved elements. They rapidly evolve until you hit birds, and then they're basically frozen. And if you take a 3-way comparison between any mammalian genomes that are phylogenetically separated, there's about 2,000 of these things, 200 base pairs or more, and almost 14,000, 100 base pairs or more, which are absolutely identical or only have one nucleotide change between any mammal. All mammals. And 96% conserved in birds. Over 300 million years. Now, Ed Rubin knocked out 4 of these things. No phenotype. No phenotype. So what do you believe? The evolutionary evidence or the phenotypic evidence? You've got to believe the evolutionary evidence because they're fiercely conserved. Something's going on here which is subject to selection that we cannot detect in a phenotypic screen, which then maybe start to think about, well, what are we screening for? Now, I think personally the history of the mammals, the vertebrate lineage, is the history of this. That, and I'll show you briefly, I'll get out of here quickly, I know I'm running out of time. Stop now? Right, okay. That's what evolution was doing, playing in the background with biodiversity by changing regulatory sequences, was climbing the next mountain, which was to introduce plasticity into the system to allow the evolution of the brain. And so when we knock out mice, we don't put them through cognitive phenotype. They can be thick as bricks. As long as they know how to copulate, and that's pretty easy. Well, it's not easy, but sorry, I won't go there, please. George Kalin in Texas is now starting to pick these things up, and he's actually got some beautiful data showing that they actually have very, very interesting behaviours if you ectopically express them. But these are mysteries, and I'd submit to you that we're not going to understand human biology until we understand these. 14,000 sequences in the mammalian genome which are frozen in every species with no explanation. There's got to be some prizes in there somewhere. I'll finish off with this because I think the evidence, and this is where we've gone at the Garvan, at least in my lab, is that it occurred to me that this hardwired RNA system puts fingers and toes in the right place. It does it very robustly, identical twins. Has then been made soft-wired by the introduction of RNA editing and modification. And RNA editing involves base deamidation. It's very highly expressed in the brain, the ADAR, and then the other one is the apibex, which do C to T. And these are vertebrate-specific enzymes, and 2 of them mammal-specific, and they show very strong signatures of positive selection in human lineage. Just very quickly, these 4 papers came out in 2004, again ignored, and I addressed a neuroscience think tank meeting in Vancouver last week, all the leaders of neuroscience in Canada. Nobody knew any of the stuff I'm about to show you, and yet they should. So editing was discovered in neuroreceptor RNAs many years earlier, thought to be an idiosyncrasy, but these 4 papers, you don't have to worry about the details, I'll summarise, showed in 2004 that, and that's over a decade ago now, that this was everywhere. So basically by comparing high throughput RNA sequencing with the human genome, they found that RNA editing is everywhere. It's mainly non-coding sequences, implying that this editing is changing the structure-function relationships of RNA, potentially influencing RNA-directed epigenetic memory. Massive increase in the amount of this in humans, 35 times more in human brain than there is in mouse brain, per gram, per any index. We're pickled in the scene. And most of this occurs in allo sequences, which, back to the dodgy logic of the retrotransposon years, were assumed to be the allo sequences, which are 10% of our genome, thought to be the last transpositional storm to hit the human lineage. But I think there's a much more interesting and more likely explanation, at least broadly, and that is that these are modular cassettes for RNA editing, which was a flexible platform to introduce plasticity in the system required for the plasticity, epigenetic plasticity required for cognition. This increases during primate evolution. I'm moving quickly because Paul's giving me the hurry up. On average, it's higher in human compared to non-human primates, and this increase is enriched in genes related to neuronal functions and neuronal diseases. I can tell you I'm skipping past this now, but there are 140 different chemical modifications of RNA that are differentially imposed. And the evidence, and it's not much of it yet, is you knock out the enzymes involved in mice, they show cognitive defects. So, and this is in the brain. So it's not just RNA editing by deamination, it's methylation, it's uridylation, there's all these chemical modifications, 140 of them, that nobody's, and there's a new field you might have noticed out there in Cell and Science and other places, articles start to appear about epitranscriptomics, not just epigenomics. So I'll skip past that. I want to just say this. Basically, there's multilateral connections between the brain and the immune system, which I think gives a lot of clues. This is really interesting. I want to finish on retrotransposon. As I said, I'd come around at the end. Basically, this is a primate-specific expansion, which appear to control retroviral and line 1 transposition, assumed to be keeping those bloody retroviruses quiet. But there's, as always, a more interesting explanation, and that is that these are evolved to domesticate these things. Fred Gage at the Salk showed that these things are moving around in neural cells in vitro and speculated they might be involved in creating the massive somatic mosaicism required for our brain. They showed this is controlled by the epigenetic regulatory protein MECP2, methyl C binding protein 2, which if mutated in humans causes Rett syndrome, a neurological disease. And then Jeff Faulkner from this institute, when he went to, and he's now back at the Mater, showed in Nature that this occurs in the brain in real time. So even the retrotransposons, who were thought to be just a graveyard of dead DNA, are alive and kicking and probably part of the dynamics of our brain. I'll finish on this. There's evidence showing that there's transgenerational reach-through, that this involves, and there's a lot of studies, a remarkable variety of factors including environmental agents, parental behaviours, maternal physiology, xenobiotics, etc., lead to epigenetic change and transmitted to subsequent generations without continued exposure that involve epigenetic and processes in RNA. And I can tell you, skip through this, but the evidence is now very powerful from two labs, from Machavi's lab in Tel Aviv and Craig Hunter's lab in Harvard, that this epigenetic transgenerational reach-through is RNA mediated. It's not chromatin mark mediated. These things are reimposed. It's RNA inheritance. This is stunning. There's this PNAS paper and this one in Nature Neuroscience, that fear conditioning with olfactory experience is influencing behaviour and neural structure in subsequent generations. So, taken at face value, these mice can remember a bad event associated with a smell. Their grandkids can. And all they've ever seen is sperm. So we can actually, our inheritance is not just hardwired DNA, but it's softwired. And there's a lot of evidence to suggest that this might be logic to suggest this might be an advantage, big advantage. I'll finish on these two furphies, and I won't have time to explain them in detail because we're running out of time, but one of the furphies is that mutation is random. It's not correct. Oops, sorry if this has gone out of order. It can't be so, and with Janet Wiles here in computer science, we got headaches every Tuesday for 2 hours. We tried to reproduce evolution in silico. Couldn't do it because the search page was too large. It's basically an NP-hard problem. And a very bright American who was at the University of Newcastle invented this, wrote a book on parameterized complexity because he realised that in any complex system, blind random searching can't do it. If you've only got a few factors, you can do it. But if you have 100,000 or 10,000 factors, it scales as an NP-hard problem. Computation, you can't do it. And the evolutionary algorithm is generate and test. So evolution must have run up against this problem and probably solved the same way that he suggested in computational space, and that is to parameterize the complex search by channeling things. The mutations are not random. Transposition patterns are not random. It never was. And it's possible. For example, if you wanted to evolve your genome and you want to play with the control sequences, you'd evolve transposons or mutational patterns that avoided exons because you don't want to damage your protein components. That's bad news. And you can see this in the transcription patterns. Sorry, the second so-called law of genetics, and this is where I return to how stupid the textbooks are. It's disappeared recently because it's clearly wrong. The so-called second law of genetics says the soma, muscles, brain, can't communicate with the germline, so-called Weissman barrier. The Weissman was an English physician, 1870, chopped the tails off mice for 5 generations. Chop. Then he measured the length of tails on the 6th generation and found that they were no different from the control set. Aha, he said. Lamarckism doesn't exist. Imagine, you miss the chop. And that ended up in the textbooks. The neo-Darwinists, and this is perhaps getting a history now, under Huxley, who's revered, seized on this when they were trying to reconcile Darwinian evolution with Mendelian genetics to say Weissman's experiment not only proved Lamarckism doesn't exist, but also that the germline, the soma can't communicate with the germline. Basically, in chopping off the tails of mice in 1870, that was the sole evidence in the textbook for the so-called second law of genetics. It's clearly incorrect. Sorry, I've got this around the wrong way. Because C. elegans proves this to be totally untrue. You put double-stranded RNA in muscle cells, C. elegans get transgenerational inheritance. Feed them on E. coli, double-stranded, goes for generations. And of course, there's been considerable advantage to be able to influence inherited characteristics based on current experience without hardwiring them. That is, to explore transgenerational plasticity. And I think this may in part be an explanation for the missing GWAS genetics, But in any case, it leads us to the possibility that our whole framework of understanding inheritance and human characteristics is actually primitive because it's so focused on AGCT rather than these things coming out of the midst which make a lot of intuitive sense. I wrote about this some time ago. For those who are interested, it has Evolution, Learn How to Learn. Sweet little article, one of my favourites. I'll finish there and say, and thank you for your attention. I'm sorry to keep you a bit longer. RNA is not the ephemeral intermediate between gene and protein. It has that function, but it's actually a computational engine of cell biology, it's a computational engine of developmental biology, computational engine of the brain, and probably of evolution itself. It's the main game. Here, there, I always show this slide, IMB and people who did the work, and now Garvan, of course, and I've mentioned some of these people on the way through, so I won't take time up by reading out the names, except to show this extraordinarily talented group of people that many of you will know and remember. Thanks very much.